And we have keynote speaker, Dr. Joel Sussman, for the first speaker. Well, Dr. Joel Sussman is a professor of structural biology and Gladys Pickman Chair at Weizmann Institute of Science in Israel. His PhD is from MIT in biophysics in 1972, and then he did postdoc at Hebrew University and also Duke University. And since 1976, he has been faculty at Weizmann Institute of Science. Now, Dr. Sussman has been made tremendous contribution to structural biology, and particularly, he has been the director of Protein Data Bank, PDB, from 94 to 99, when he was at Brookhaven National Laboratory. He solved many structures in key proteins in the nerve signaling pathways. Today, he will be talking about Proteopedia. It's a scientific Wikipedia where, well, people can contribute and share information. So, Dr. Sussman, please.
Thank you very much, and I want to thank the organizers for organizing the meeting and inviting me to come. So what I want to talk about is just what's described, Proteopedia, and maybe I'll begin with the very simple slide of a young girl, but it could symbolize anyone. Don't take it as a gender specific. And I wrote down, it's hard to believe, but 3D structures are often hard to understand, even for structural biologists. And I think this is a major point. And I think maybe in particular for structural biologists, and they often go out of their way to describe every single detail of a structure. And if you hear a talk by a structural biologist, not all, but some begin with the 15-minute introduction. What is the problem they're working on? And then they say, oh, and we're going to solve this structure. They show the crystallization, they solve the structure. And many, many beautiful pictures, and they forgot to answer how the structure relates to the function. And this is really what Proteopedia is about, to try to relate the structure to the function. So I'm going to give several examples that I've made, animated pictures of what it feels like to use Proteopedia. And so you'll see them maybe starting with the first one. So this is a page on the HIV protease where you're sitting on your PC or Mac, and you go to the website, and then as you read the text, you get to these green links which describe something about the structure. In this instance, it says that the HIV protease is made up of two symmetrical subunits with a hole in the middle, where there's some residues that are playing a key role. And every image, as you go, click over here reading the text, gradually changes the picture from the previous one to the new one. And it says there's even a tunnel looking down the structure, right through the middle of the HIV protease. Of course, this is a protease which is the target of many of the drugs for treating HIV. And here it says, as you look at this, the whole protein isn't static. And this is crucial, of course, as many proteins are moving. In this instance, it's showing a simple morph between the structure of HIV protease in the absence of an inhibitor or ligand, and then when the ligand binds, there's an enormous change in these flaps. These flaps are crucial for the way in which the protein binds to the HIV inhibitors. And without knowing the structure of the protein this way, you can rotate the pictures with your mouse anytime you like. You can see how these residues don't just come down, but the side chains and loops change their conformation to bind to the inhibitor. And then we go down and describe some of the inhibitors that are found that are used for treatment of AIDS. For example, sequinor or inverase. It shows the picture of the drug by itself. And then we can see the drug bound to the enzyme and how it affects the enzyme. So there you can see it bound tightly inside the enzyme in exactly the hole that was described previously. We look at it You can blow it up, and each time you can jump around anywhere along the text, forward and back, grab a hold of the images, and so on. So this is one example. In this case, we can look at different drugs and watch as the structure comes down, binds the inhibitor, APO, bind to the inhibitor, and so on. And these morphs we find, for myself, extremely useful because if you have two pictures, one next to the other, sometimes it's not as clear as to show the change in conformation, even if it's just a kind of schematic change. And then you can see a series of different inhibitors bound to the active site. You'll see that they're different, but they all have some similarity in ways which they bind to the glutamic acid residues, which are crucial for the catalysis of breaking up the polyprotein in the HIV infection. So this is one case. At the bottom, you can see the names of the people who contributed to this page. And we'll come back to this over and over again. This is one of the major differences between Proteopedia and Wikipedia, although we're using a lot of the tools of MediaWiki with permission from MediaWiki, but we insist that you register to make a page so we know who does it and you get the credit. So everyone knows that the PDB is increasing enormously. I was here early days, but now it's jumping up to almost 100,000 structures. And you can find pictures of proteins very often on covers of some of the top journals. This is one we'll come back to, one of the structures we've worked on, the acetylcholinesterase, and I'll give some examples of that in Proteopedia. But again, looking at these pictures I think are fun. You often put them on your walls in your office, but until you read the text and relate the text to the pictures, they're just pretty pictures. So this work was, I think really actually, the idea came from me, but I want to give full credit to young student Aaron Hodis and Chaim Pruski, the head of bioinformatics at the Weizmann. Aaron was a rotation student and then a master's student with me when he developed all of this idea, initially on a Mac, and then he put it onto a Linux box with permission again from MediaWiki to make all this. We added in this three-dimensional JMOL, which is a public source, open source viewer for web viewer, and then Aaron developed an extremely nice front end for JMOL. So the easiest part of making the page are making the figures. That comes out easy. Aaron left. He had a terrible tragedy in his family and returned to the USA, and now is at Harvard, MIT, doing his PhD jointly in the two universities in Boston and Cambridge. So this is a typical picture of hemoglobin from Proteopedia with the hyperlinks showing how the pages change until you get down to here, showing the sickle cell anemia mutation. So I'll give you a few examples from Proteopedia. 2009, the Nobel Prize was awarded to Venki, Tom Steitz, and Adi Yonat for their determination of the three-dimensional structure of the ribosome. So we have the entire ribosome on one of the Proteopedia pages made by Wayne Dakuta, a postdoc in New Hampshire. And this is a picture of what it looks like if you look at the Proteopedia page of the ribosome. This is the full 70S ribosome particle blown up. And under it, Wayne put some tags, and it's quite interesting. This is the whole structure with the nucleic acid and the proteins. He turns off the tRNA, messenger RNA, and the ribosomal RNA. And what you see is something which has almost never been published in any journal, is the proteins are floating around in space. In some sense, the proteins are just decorating the RNA. This is the RNA structure, and if you didn't know it was RNA, it looks like a protein structure. So this kind of thing quickly can give you some information that you might not have seen in any of the articles. This is one of the subunits, and then the other subunit of the ribosomal RNA. And this can be easily loaded without any additional software on your computer. In addition to structures, we thought it would be useful that Proteopedia might be used as an online mini textbook for structural biology. We got permission from James Holden to show some pages of what different resolution. So you can see over here, low and higher resolution down here and lower resolution there. But it's much nicer if you look at it as an animated picture going from highest resolution, which are the smallest numbers, to the lowest resolution, which are the higher numbers, 2.8, 2.9. As you get to higher and higher resolution, going back and forth, you can see how much less detail you have. Then it will cycle back again. When it's really the high resolution, you can even see the holes in the tryptophan ring here. So we have many, many pages starting from amino acids up through secondary structure, hydrophobicity, intrinsically unstructured proteins, many topics which are now being organized as a mini course on the web for people to learn. And these are all contributed not just by us, by people from all over the world. So this is another example, one made by Eric Martz and Aaron Hodis, comparing two papers back to back that appeared in Nature in 2008 on proton channel. And both papers were excellent. Both papers referred back and forth to the other paper. So they knew they were coming out together, which was very good from two different groups. One was an X-ray, one was an NMR structure. But what's important is that one of them showed the open conformation, one of them showed the closed conformation, which is the way in which protons would go in through the cell membrane. But in none of the figures in any of the articles did they compare the two. So what Eric and Aaron did in this case is they took the two structures and they made a morph showing the comparison between the open and the closed. They showed it as schematic, they showed it as a space filling, and then they went in to look to see which are the key residues that are actually closing up and opening up the channel for the protons. In this instance, they can see it as prolines over here. It goes tightly closes up and opens up. And so this is a really nice example of showing how what's important often is not a single structure, but a comparison. And if you go to hear a lecture from top structural biologists, often they show movies that they make, and so the talks are often really, really good, but when it gets to the printed articles and even the supplementary articles, supplementary information, often you don't see the nice links between the text and the information. So at the bottom of each page, you can see Proteopedia page contributions and editors. In this instance, the names of the scientists or students who made the page. You can click, for example, we can click on Eric Martz here, and you get his page. He actually made a page about himself, a little bit like Facebook, but not the same. I didn't want to pretend. He was giving a tutorial in Osaka showing physical models, and this is Eric himself. And if you look over here, there's a little page saying email user. And if you click on that page, because when you register, you have to give your real email, and we triple check, and your real name. You click on that, and you can mail a message. So I was typing a message when you were on the trip today to Eric. Dear Eric, recently I was looking at your and so on, and then you just push send. And so most importantly, though, not just for someone whom you know, let's say you were looking at a Proteopedia page and you saw a terrible error. You could try to write a nice message to the person saying, I think I saw an error, or you could go in yourself and correct it. But it would be a way to communicate with the authors. It's not like in Wikipedia, which you don't know who has written it, so all you can do is just modify the page. This is another page, which is really dear to my heart, and maybe to Keith as well. It's a picture of the DNA binding region of the Lac repressor. Lac repressor is one of the crucial proteins that you study in early stages of molecular biology that turns on and turns off the Lac operon. And it was one which defied crystal structures for many years, and then it was determined in the lab of Lewis and Ponzi Liu in Philadelphia, and then Robert Kapteyn determined the NMR structure. In Lewis's lab, they determined the structure of the Lac repressor DNA binding domain bound to its cognate DNA, the DNA that recognizes on the operon. And Bob Kapteyn's lab in Utrecht determined the structure of the Lac repressor bound to the non-cognate, to a random piece of DNA, sort of when you could think of the protein getting on the DNA and sliding along. And what he found, and this is a picture showing the NMR structure, and still I didn't make a movie here. In this instance, there's few helices that are bound in the binding groove of the DNA, and a huge amount of disordered tails. So in the NMR, you see an ensemble of all these different structures. When the protein moves along the DNA and hits its cognate, the proper signal portion, these tails suddenly become structured and lock into the DNA. And so we've made them. There's a whole series of individual pictures on the page, but towards the bottom, we have one picture showing this change. This is the cognate, uncognate. So here it's all flopping around. We just showed one of the pictures. This is all many different conformations up there. But you can see this portion over here turns into a helix, really changes its structure. And not only that, the portion of the DNA it's binding to is not a simple straight double helix, but sort of a kinked helix is what they saw in the laboratory in the X-ray structure. So these are the kind of examples over and over again that you see that it's crucial in structural biology to be able to see not just one structure, but the changes in structure. And that's in some sense, I think, why, in my opinion, why proteins are so much larger than small molecules, because they have to be able to change their conformation to do something. And so this tool is a nice tool. On this page, again, I want to stress, I'm showing all the pictures. We built the tool, but the pages were made by people from all over the world. And this is by Eric Martz in Massachusetts. But of course, it looks like only a geek could make this. Although I remember when we first presented this, Aaron gave a lecture in Erice in Italy in a workshop like this. And at one point he said something like that. He said, but you know, even a professor could do it. He invited me up to show how to make a page. So in this instance, I will not show it. I'll show some other people who did it. These are in case Karl Oberhauser and Messiah College gave assignments to undergraduates in his course he was teaching. And each student would have to make, after studying, they didn't do it quickly. They studied a protein for a month or two with his help. And then they wrote up a text and pictures, and then they made a page. And this is one case, an undergraduate, Miss Emily Foster, made this page on the photo system too. I won't show the animation in this instance, but at the bottom, Emily's name appears, and I've been in touch with Emily since this, and she swears that she was accepted at university for graduate school and nursing school only because of this page. I think she was trying to make me feel good. But she got both the credit, and I think it's this, well, it's important, the responsibility. So when you make a page and your name appears on it, you try every effort to make it right. So how do you get to make a page? Anyone in the world, the site is free, and for both making pages and viewing it for companies and for students and scientists. But if you want to make a page, you have to request an ID. So on the home page, you request an account over here. You're not logged on. And then you get a page where you type in your name, your address, your university, and a little bit about yourself so we know. And then, like anything else, you get sent an email message where you confirm it's you who did it. So if you try to apply for an account, make sure that you give your full name and so on, because it's not checked by a computer but by a human being, because we want to try to regulate the people. We have over 2,400 registered users in 45 countries now contributing to Proteopedia. So how do we judge if the page is right? We've set up a Proteopedia editorial board with John Mould, Chaim Produsky, Eric Martz, Wayne DeCouter, and several other people. Israel Silman, who's an expert on proteins of the nervous system. David Kanner, who's at Memorial Sloan Kettering and now at MIT, and so on, who have done a lot of analysis. Every page is checked, and if we find a problem, we try to write nicely to the authors and almost always people are happy to correct the errors. So this is not just used in universities and graduate schools and scientists making pages, but there's a daring person named Tim Herman from Milwaukee who has set up a National Science Foundation project in the United States to have what he calls smart teams, student modeling a research topic, where he has mentors from universities, either professors or graduate students, coming to work with students in high school over the course of months to get them excited about structural biology, building usually initially physical models, making a nice poster like the posters in the back here, which often go to conferences, and then to publish it in Proteopedia, which is sort of the end for them because it's really like a publication. So I'm going to show you one such page from a group of students in Milwaukee. And this is a page on the beta adrenergic receptor. This is a very important protein. As probably everyone knows, Bob Lefkowitz and Brian Kubilko were awarded the Nobel Prize last year for this really momentous achievement, because I think 30 to 40% of drug targets actually bind to this fold this seven helical membrane structure. And actually, when this page was originally made, they made a terrible mistake, in my opinion. This was before the Nobel Prize was awarded. They made it 3 years ago, these students. And they actually made a video clip with sound, which you'll hear in a second. And they only mentioned one lab, and neither of these names was mentioned. And I didn't have premonition because there were several scientists working on it, but I wrote to them back. I said, look, I think for political correctness, you should mention the other labs. And I would say within an hour and a half, the video was changed, and they corrected it themselves. They were not too happy. So this is a movie taken from the page of the beta adrenergic, made by high school students. And so let's see if it works. This got sound.
This is a physical model of the beta adrenergic receptor based on the 2RH1 PDB file from the Kobilka, Weiss, and Stevens labs. The beta adrenergic receptor is a member of a large family of proteins known as the G-coupled protein receptors. The protein spans the membrane, which would lie in this horizontal orientation. This receptor is designed to specifically bind adrenaline, which enters here from the extracellular environment. Upon binding, the protein undergoes a conformational change that results in a signal being transduced through the membrane, where bound G proteins propagate and further transduce the signal. This is an alpha carbon backbone model. As a member of the family of G protein coupled receptors, it has the characteristic 7 transmembrane alpha helices, colored green in this model. We have displayed a beta blocker bound in the active site of the receptor, surrounded by several amino acid side chains that contribute to the specific binding. A particularly interesting feature of this protein is the extracellular loop number 2 that connects helices 4 and 5. 2 disulfide bonds, shown here in yellow, hold this loop back and pin it to the top of the structure, opening up the channel that allows the diffusible hormone to enter the binding site. We've also displayed 3 cholesterol molecules that are bound to the outside of the receptor. These are thought to be involved in the dimerization of the receptor in the membrane.
And it goes on down to show pictures. All of these are interactive links and so on, but I think the movie is really by itself wonderful. This is a picture of the students where they made their poster and so on. And then they have their names at the bottom where it was made at this Smart Team project. I think in this instance, when I first started talking about how can high school students talk about something so complicated, but it reminds me a little bit of the old children's story of the emperor's new clothes. Sometimes kids can see things that the parents don't see. In this instance, they really, I think, described the structure as an introduction. I sent this page to Brian about 2 days after he got his Nobel Prize. He wrote me back within about 15 minutes. He said this was the best description he's seen ever of his own work. And it was a nice feeling to hear him say that about what students had done, high school students. So this is another case. This is a group of students that I've been working with in the South Bronx. It's actually a gifted bunch of students who are really good, but I think in particular the teacher, Miss Alison Granberry, is dedicated. It's a small group that comes in once a week for the entire year after school to study protein structures. And to get kids to do that instead of playing Facebook or playing football or something is impressive. So the EMBO Encounters, a magazine in Europe, wrote this up as more than just pretty pictures. And I think this really captures what Proteopedia is about. It's not just the pretty pictures, but to try to describe the structures. They worked on a protein that we had studied. That's how Alison got in touch with me. She didn't know that I was involved in Proteopedia. She just wanted to make a page on acetylcholinesterase. And they made physical models, and then they showed how a snake toxin bound to the enzyme, blocking its activity. And the students really learned a lot. This young woman, Marissa Van Brockel in particular, she's holding the model of the protein cholinesterase, wrote to go to Union College as an application and applied for a Gates Millennium Fellowship. And she put on her CV again that she had made. She was the first author in this Proteopedia page. And she got into university and she was awarded a fellowship which covered all her undergraduate and graduate student fees and housing. So she's very happy about all this. So I think, but more important, I think they learned a lot in all of this. There's another thing about Proteopedia which I think is important, which we began about a year and a half ago. And this is together with the number of journals which are listed. These are the ones so far that are working with us. When an article is published in a journal, often there are very good figures in the journal, and very often nowadays there's supplementary information, which often includes extremely good movies of protein structures which help explain it. But nothing really which links the text to the figures like in Proteopedia. So these journals from Cell on down to PNAS and proteins and so on have agreed to let us work with the authors once a paper is accepted and to build with them a Proteopedia page which is embargoed until the paper comes out online with the permission of the journal. So simultaneously, a short page in Proteopedia comes out with a hyperlink, of course, to the online version of the article. And in the article, there's a hyperlink back to Proteopedia as a way for the readers of the article to read the article. And if they ever want to see any of the pictures in the article jumping out in 3 dimensions with the text, they can go to Proteopedia. So I'll show you an example of this in the Journal of Biological Inorganic Chemistry, which I'm sure everybody in the audience reads every month. And so this is an article on anti-cancer ruthenium half-sandwich complex. And you can read what it would look like. This is how it might look like in the text if it were arranged this way with the structure over here. And you can go through, and this is what it looks like in Proteopedia. So you start reading about the organo half-sandwich, and immediately it lights up. And it lights up with the background of the way it looked in the first pictures. You can see where the molecules are fitting. And you can go through, and you can see that it's bound in a hydrophobic interaction, a series of aromatic and hydrophobic residues sitting around this region. And there's different ways of displaying it, so you don't have to just see it in one way. In the second, you'll see that there's the sandwich between these regions over here. And it goes through, and it's rather short. The articles are almost in the spirit of what you'd see in Nature as a news and views, but it comes alive in a way that's different than just a printed text. So most of the articles are written at that level, or the level, let's say, of the way Scientific American often writes articles. So how are these interactive 3D complements made? So the editors of the journal alert us that an article has just been accepted. They write to the author that they will be contacted by the editorial staff of Proteopedia. And what we do is we write to them. We have a simple web page, actually, for them to fill in the title of the article. We need the email of the contact person, the unique journal, paper ID, and then we go to this hyperlink, which only needs a few bit of text, 2 or 5 images of the structure, the PDB ID or coordinates if it hasn't been released. If one sends us some PyMol, which is a very commonly used software, we've written a tool to convert PyMol to the interactive web JMol program so we can use this. And usually we like to have the manuscript. And then we have scientists at the Weizmann who go over this and basically write the equivalent of a news and views on one page. We try to make the pages really rather short with the meat of the article, so it won't steal from the article of the journal, but really complement it. And this is crucial. The Proteopedia Interactive 3D Complement is embargoed until the paper is properly released with the blessing of the editors of the journal. So this is, I think, some direction which I think would be wonderful if more journals were to adopt it, because I think it would make the papers on structural biology have a lot more impact to the structural biologist and, more importantly, to the non-structural biologist. So this just one last topic, which is a topic dear to my heart, is some work that we've done, and I'll show you the pages that come out, some other kinds of examples that how Proteopedia may be able to help, not just in the English language world, but beyond. This is on some studies we did on the most common current therapeutics for the paleotropic treatment of Alzheimer's. That's the treatment of Alzheimer's in the sense to slow it down, but it does not yet cure it. And it's based on a paper from 1982 from Bartas's lab, which said that if you can somewhat increase the cholinergic activity in the brain, which means to increase the levels of acetylcholine, he showed in a large number of studies, and animal studies with mice, that this tended to improve their memory. This was the real basis of all but one of the current therapies for treating paleotropically Alzheimer's. These are called the cholinesterase inhibitors. And we would like it if it were more in the spirit of the conference to take 2 genes and call me in the morning, but it's not there yet. So there really are small molecules. These are natural compounds that you can isolate from plants. These are synthetic compounds. This is what acetylcholine looks like. It's quite amazing to me, having said lots of chemistry, that these look so different from acetylcholine, and they all bind virtually in the same site as where acetylcholine binds. So in some ways, it's nice to be able to compare these. I just want to very briefly show you in Proteopedia this molecule, which is the most widely used drug. It's a several billion-dollar a year drug called Aricept. It's widely prescribed. And this one, which is a natural product, it's actually a traditional Chinese medicine, which is an approved drug in China and is sold in health food stores in the United States because it's a natural product. It doesn't need a license for treating Alzheimer's. So that's the Aricept. And this one over here is called huperzine A, which was actually discovered in Shanghai. And we'll go to it in a second. So this is a picture of the page of the Aricept page in Proteopedia, showing Aricept bound all the way along the active site gorge. This is the entrance of acetylcholine, and down here is where acetylcholine is broken down to acetic acid and choline. And we could look at it. It shows that Aricept binds in a very unique way from the very bottom to the very top of the gorge. And it quickly shows this. So if you're looking at the article later on, you'll see it. This is the first key binding area. There's an aromatic residue that binds at the bottom, tryptophan 84, and the top is another aromatic, tryptophan 279, way up there. And you can see where they're labeled in the figure. The Aricept was discovered or synthesized by Eisai, a Japanese pharmaceutical company, and sold by Pfizer now as a joint project. What's particularly interesting, though, is that the Aricept binds all the way along from the bottom to the top with a sub-micromolar binding affinity with not a single covalent or hydrogen bond. All the interactions are either mediated through water molecules, which is written here, or through pi-pi interactions between parts of the structure and the aromatic residues. So it's very specific, but it's nothing which is binding in a normal kind of way that docking studies would show. And this page kind of shows that nicely. So, but that page is in English. But there are other languages besides English in the world. So this particular page has been translated to French, Spanish, Arabic, Italian, Turkish, Chinese, and Russian. And so this is an Arabic page made by Monif Ayash at the Weizmann Institute, a Palestinian student studying at our institute, and he kindly offered to translate the page into Arabic with all the hyperlinks down here. I don't want to show movie because it's just the same story as before. This is the table of contents in Arabic showing the different portions. This is the huperzine, just to give you some background for the Chinese in the audience, they can read it. It comes from this Chinese moss, Huperzia serrata. It's the leaves of the moss that are put into tea. That's how it's given as a traditional Chinese medicine, and you drink it, and you drink it hot. So some of what's in the leaf will come out. And Professor Tang and Professor Xu in 1986 did very careful chemistry and found one active compound in the leaf of this Huperzia serrata, which is this molecule here, huperzine A, which they found was a potent inhibitor of the enzyme. Y.P. Pang was working at the Mayo Clinic, and he contacted me right after we determined the structure of the enzyme and suggested we determine the complex with this to see if it bound any way like the Western compounds. And Mia Rabas, a young Dutch graduate student in our lab, determined this structure. And it was published in Nature Structural Biology. And this is a page of this translated, of the huperzine Proteopedia page translated into Chinese by Dr. Yichun Zhu. She was a postdoctoral fellow in my lab and now has returned to the Shanghai Institute of Materia Medica in Pudong, where she has a really nice laboratory set up. So this is a short page to show you what it looks like in Chinese. I can't read it, so I will need the help of you, but if you can see the text, you can look. This is obviously pointing to the huperzine molecule. When she clicked on it, suddenly the huperzine itself lights up, and you can bring the mouse over there. I'm sorry, it jumps because of the way I animated the thing, but you can actually rotate the huperzine around with the mouse. So you can make it bigger or smaller by just using the tracking pad of the mouse. Then you can come down here and see some more text. And what Yichun tried to show here, this was part of her PhD thesis while she was in Shanghai, was that the structure is bound by a whole series of residues around it at the bottom of the gorge, very much like the Western compounds. So she first colored them all green, and then she highlighted them with a different hyperlink purple, and then she goes through and then isolates each individual kind of interaction to try to show the specificity that nature somehow chose. It's possible that this plant evolved to have this huperzine in the plant as a natural insecticide because, in fact, I think as many people know, inhibitors of acetylcholinesterase are very potent insecticides, and this possibly was done that way to keep the bugs off the plant. So this is the active site, and the second then she will click on one of these. The serine is the active site serine over here, and then you'll see where the huperzine comes into this picture. These are the residues, part of the active site, the histidine, and say that's the huperzine coming, showing where it binds. And then her name is at the bottom, but I will go a little bit forward. But I think it's useful to have pages in other languages, especially for younger students who probably are unable to read English as well as the older kids. So coming soon to Proteopedia, which we're working on, we're going to have very soon not just the structures from we made a lot of automatic structures from the protein data bank, which is our main basis, of course, of how we get the information. It's still a wonderful resource. But sometimes crystallographer determines a structure where it's an individual structure, but in fact, the structure is really complex, like hemoglobin is a tetramer. Often it's determined as a dimer. So one would like to know what is the natural biological unit. We are working with a group in Germany to do a series of chemical reactions. The first set are the entire Krebs cycle. We will show the chemical reaction as animated movies together with what happens when the enzyme is present. And this is work in project. Other major problem we faced, which probably no one in the audience would know immediately, is all of this is based on the open source JMOL, wonderful viewer which has all these options to show things, but it requires Java. And Java has been declared a security risk by the TSA in the United States. And every so often they send out a way to turn it off all PCs and Macintoshes until Oracle comes out with a fix. And so this cannot continue. So working with Robert Hansen, who came to visit our lab, he's the main developer of JMOL. He's created Java ScriptMol, JSMol, which now works on even iPads, which of course don't have Java. And we hope to make this work a lot faster. It works somewhat slower than JMOL, but at least it's running. We would like it to run on all tablets because I think tablets will be one direction of the future. We have set up with Nir Ben-Tal in Tel Aviv with a single click, if there's multiple sequence, it shows immediately the conserved regions in sort of dark maroon and the unconserved region in bluish, so you can see the active site. So this comes out for almost all of the proteins in Proteopedia. You could click on that and see this automatically generated with the collaboration with Nir Ben-Tal's group. And this was published recently, both the JSMol and the evolutionary conservation was published on the front page of the Israel Journal of Chemistry as a special review issue that Chaim and I were the editors for. So just in conclusion, we need your help because I look at this, maybe there'll be other tools, I'm sure there'll be other tools coming out, but this is an example, I think, of one way for next generation publishing to begin to think. And it would be nice if people would add to it. It's useful to make structured tutorials and lectures for classes. There's an option, although most pages are open for anyone to edit, there's an option where you can lock your page so that if you come back next week, it will be the same, no one can change it. So if you made a tutorial for your class, you have that option. It's really an example of crowdsourcing for scientific annotation. Currently, there's over 97,000 pages from over 2,400 registered users in 45 different countries, and it's growing. But I think without you, Proteopedia is just an interesting tool, but unless we get really good content, I don't think it's really very good. So these are the people who worked on Eric and Chaim are the main developers. Aaron and Chaim. Eric Martz, who really is one of the great figures on using web tools for structural biology, spent a lot of time in Israel. This was our main laboratory, was this Irish pub right near our institute, and it's called the Dublin Pub, which is very nice. And so we thank you, and I thank all of the contributors of Proteopedia, students, teachers, scientists all over the world, as well as some of the funding agencies. So thank you very much. I have put outside postcards of Proteopedia, so you can take one or use it to send to your friends. All right.
Okay, now we have time for several questions.
Yeah, so I'm Dongxi from University of Missouri. This is really a wonderful website. So my question is that, do you plan to accept some modeling results, for example, some molecular dynamic simulation of protein, or maybe using structure prediction to add a loop for the missing part of the protein?
Yeah, the answer in short is yes. We make sure that when it's a model, a strict model, that it's written on the very top of the page. This is a model, so everyone knows about it. The PDB made a decision several years ago not to accept models anymore. And my feeling is that there's really a fine line between a model and an experimentally determined structure. And in fact, many structures are now using a combination of many, many different methods, from SACS to EM to particles from the X-ray parts of it, putting it together. So in some sense, those structures are called experimental, but they kind of go between experimental models, but we accept also molecular dynamics, simulation models as well. But they're labeled.
Wonderful system. Just a question.
I don't hear you.
Okay, you can't hear me?
Microphone, please.
Oh, okay. Yeah, now? Am I audible? Okay, still not?
Go ahead.
Wonderful system, Joel. Just a question. So, for example, coming from a genetics background, we know coding changes which render a protein non-functional, but we don't know how it does that, for example. So is it possible to just take such examples from strains and at least predict that this is the known structure and what could possibly be happening?
Yeah, it's a very good question. We want to do that. We haven't done it yet. One of the people on the editorial board is John Malt, and he's spent a lot of his time in the last several years working on looking just at this topic for his research. He's looking at SNPs to try to understand the effect of SNPs on 3D structures. But it would be nice if one could make this in an automatic way, at least to show where the SNPs are and to say what is known. I think one should be very cautious about saying if the SNP is just a simple replacement of the amino acid, or if in fact it may result in a conformational change unless this were known experimentally. But the idea is, I would like to do it, we just need a little more manpower and someone who knows how to do it in a way that's transparent. Good idea.
Yeah, that is a good idea. I didn't think about it. Some of the edgeotyping that I talked about yesterday might actually be sent over to you guys. You could put that in. We can discuss this offline. My question was going to be, are you planning to put data from so-called structural proteomics or structural genomics sort of systematic efforts, as we, I think, still have them in the States? I'm not even sure how. You see where I'm going.
The answer is completely yes. What I didn't show, I only went 3 minutes over time, but I didn't want to go any more. Of those 97,000 pages, 93,000 of them are automatically generated pages by a robot tool that Dr. Chaim Produsky developed. The page looks like a perfectly normal page, which has abstract of the paper and many hot links. Many of the structures of those 93,000 are from the Structural Genomics Initiative in the United States and throughout the world, but they're labeled often, and this is very important, often those structures don't have any papers published. So the links, the hyperlinks that are put into this automatically, where it's linked to the gene, it's linked to the PFAM family and all these other points, all those are there available, but of course at the bottom it's labeled which structural consortium determined it, because that you can get from the PDB.
Hi, wonderful talk. I was wondering if, suppose I have some observations on some functions, and I don't know what protein or proteins might be performing that, or what's the structure of those proteins. But I input the description of my observations, and I can approximately get the kind of structures, because you are mapping structure and description, right? So if I can search based on descriptions, can I get the approximate structures that might be able to possibly performing this?
You're pushing things, but the answer is maybe. What we did is we have two search boxes. One is an extremely explicit search box, which will be the name of a protein or the PDB accession code, the ID code. We also have a Google search within Proteopedia. So the latter often will pick up what you want, but I don't want to guarantee.
Possibly the protein has not been solved. I mean, or we don't know, it might be a novel protein. So can it help me predict the structure for that?
Yeah. Right now, we didn't implement that primarily because there are many other servers around which are really well-developed servers for protein prediction. We have pages in Proteopedia, if you went there, to tell you which are some of the commonly used ones. We don't have that. What we are going to do, though, is we're working with colleagues in Yale who have a wonderful morphing server to make it easy that if you have two structures to be able to go into Proteopedia, it may send it to Yale and come back, and you'll be able to morph. But I don't know if we're planning right now to make a protein prediction, because they're really good ones, and I don't want to try to reinvent the wheel.
Can you tell your name and affiliation first?
Yeah. Hi, Daniel Weizmann from Tel Aviv University. Can someone build a tool that converts all the PDB entries into general pages that someone can later edit?
Yeah. This is precisely what Chaim Produsky developed. We call these seeded pages. So every week on Wednesday, as you probably know, the PDB comes out with the release of new structures. By Wednesday evening, all those pages appear as standard format pages, ready to be edited by people, but they have a lot of information before that, and these all exist. The answer is yes.
Okay, and this is look well like a wonderful site. I will definitely use.
Including this work of Nir Ben-Tal from Tel Aviv University, which has all the evolution. All of his evolutionary information is automatically generated back and forth from Tel Aviv to Rehovot.
Okay, great. Thank you very much.
All right. So
if anybody wants, I put a few of these outside. You can pick up.
let's thank Dr. Sussman again. Thank you very much.